walk behind me because otherwise there will be an accident. Okay. So thanks, Jikun, for the warm introduction and for inviting us all here. It's a great pleasure to be back here. I was here 4 years ago, but nobody of you can remember that. So let's get started. So today I'm going to talk about some recent work that we have been doing in analyzing really large images. Okay, and I'll explain what I mean about that in a minute. So this is joint work with a number of PhD students in the computer vision lab at SUNY University and a number of collaborators in the biomedical informatics department and ecology and evolution. So you'll see that this is very interdisciplinary work, and since the theme of the day here is AI, this is actually kind of an example of how AI is getting into a number of different scientific fields and is actually making an impact. So I will mention two applications here, two different scientific domains. One is what is called histopathology image analysis. Do you guys know what biopsies are? Okay. So when you go to the doctor and they want to look at a piece of your skin, or if they are worried you might have cancer, they take a piece from inside you, from an organ and so on. They take a very tiny piece of tissue and they look at it under the microscope. Okay? And this is a process that has been happening for more than 100 years now, since the end of the 19th century, and it revolutionized medicine. A lot of lives were saved. This is how even today, this is how cancer is diagnosed. A particular doctor who's called a histopathologist, or just to be a little shorter, a pathologist, is looking at a biopsy on the microscope like this, shifting left and right, until they are sure they see enough signs of what they think is cancer, and they decide what type of cancer it is, what stage it is, and so on. Why is this important? Because based on that, the doctors later will decide the treatment. They will decide if the patient will get an operation, if they will get radiotherapy, if they will get chemotherapy, what kind of chemotherapy, what kind of drug, for how long, and so on. These are very important decisions. If you get them right, the patient has a lot of chances of surviving. If you get them wrong, so this is how this has been done in the last 100 years. But now, what's happening is that these images there are scanners where you can take the piece of tissue on the glass that has been chemically treated to look at the microscope. There are scanners that will actually take an image of it. And this image is not the size of the images that your cell phone takes. I know that the Korean companies now make cell phones with really high resolution. But here we're talking about images that are about 100,000 by 100,000 pixels. Okay? So Samsung's not there yet, at least not on the phone. They might be making scanners, but that I don't know. Okay? So now, for the first time, we can actually do AI on those images and try and make automatic analysis, not to substitute the pathologist. There is no way at this point that we can get a human out of the decision process of what cancer it is. Nobody would trust a machine to tell them that they have this kind of cancer and they have 6 months to live. They would always want a human doctor to listen to it. But to actually give more information to the doctor to make the decision, to That's one thing that is interesting. The other thing that is interesting is that now we can actually answer questions which were not possible to answer before, because the doctor can only look and form an impression. But if there is 100,000 versus 500,000 or versus 1.2 million of a particular cell type, and that means something for the diagnosis and the prognosis of the cancer, that is something that the doctors couldn't do. Now we can do it, and now we can help doctors find new ways to characterize cancer, new ways to describe it, new ways to find what might cause it, what might affect its development. Here's another example of very large images. Okay, we've worked with people in Stony Brook who are doing counting penguins. Okay, and the way that why is this important? Because knowing how the penguin populations are doing is telling us a lot about what is happening in the Antarctic, and probably you guys know why we care about the Antarctic now. We don't want it to melt. It's kind of a snapshot for what's happening to the planet, right? So they can they go there. The penguins live in the same spot every year. Okay, they come back every year. They have their eggs. They have their families. They stay on the same spot. In the past, the scientists had to go once a year or once every couple of years with a boat to the Antarctic. It's kind of cold. It's kind of expensive to go there. And they would stand in the middle of the colony, and they would count with a clicker, just like this. Okay? And they would count 80,000, 90,000. We're talking about really big numbers. And clearly, not very accurate counts. So now there is enough satellite imagery that they hope to do this with the satellites. But listen to this. The penguins are still too small. You can't see them from the satellite. They're smaller than 1 pixel. Okay? So the smallest thing you can see in an image is a pixel. And on the satellite, a pixel is about 30 by 30 centimeters in the ground. It's approximately as what I'm standing. Okay, I'm a big guy. You could probably see me from the satellite. But a penguin like this and like this, you probably won't see it. So how do we figure out how many penguins there are? Well, guess what? And guys, please don't laugh. The penguins stay there half the year. Okay, and they don't go too far. So they're birds, and they create droppings. Okay? And these bird droppings change the color of the earth. And it becomes a little bit, you can see it becomes a little bit redder. Okay? And so what we're trying to find is this red area. Okay? And decide, has it shrunk? Is it bigger? Is it smaller than last year? That means that the number of the penguins is going up or it's going down. Okay? So these are two problems that I will use to show you how you can do artificial intelligence. It turns out that the two problems are quite similar. Okay? The satellite images and the histopathology images are quite similar in what it is that you are trying to find. So, of course, you guys have heard of deep learning, of convolutional neural networks. These are the really strong algorithms that have been very fashionable in the last 5 or 6 years, because they actually do a really good job in a lot of tasks, and that's why we're talking about the AI revolution now, because there's so many tasks that we can solve that we couldn't solve before. Okay, so these are some very standard examples. You can recognize handwritten digits. You can recognize traffic signs. You can recognize street numbers. You can even take a picture and decide what is every pixel in the picture. Is it a building? Is it the sky? Okay. So this is stuff that has been happening, as you can see, for the last 6 years or so. You can recognize faces very, very well now. Okay, and for the older people here, you might have noticed how your phone or Facebook now are very good at recognizing you. For the high school students here, you just know this since you remember yourselves that the phone can recognize you, right? So, a very important thing in the field of computer vision and artificial intelligence was a data set that came out in the beginning of this decade called ImageNet. This data set was trying to collect images for every category of object as you define it in the dictionary. Okay? So some linguists went and collected and created a structure of all the words, of all the nouns. Okay? Things like mammal, placental, carnivore, canine, dog, working dog, husky. Okay? And these are all different categories. Okay? And they collected images for all the bottom categories of the tree, like all the huskies, all the bulldogs and so on, and then they had pictures for all the dogs, and then they put in the cats as well, so then they had all the carnivores, and so on. So they created this humongous data set that looks like this. This is the images that they had for the word hammer, right? And then the computer vision scientists were trying to actually recognize things out of those images. Okay? And what they were trying to do was train classifiers. I don't know how many of you know what training means, but this is the idea that you have a program that takes in the images, knows what the correct answer is, it knows whether this is a hammer or not, and modifies its parameters so that it can actually predict as accurately as possible for these images whether it is a hammer or not. And then, if it's successful, what you hope is that any new images for which you don't know the answer will behave approximately the same, so your program will give you approximately this accurate prediction. So ImageNet was a big contribution because people could do very, very large scale experiments, and for a while it was really hard. They couldn't do much. And then in 2012, there came out this paper that actually blew the competition out of the water, okay, on this ImageNet challenge. They could do much better in recognizing things. And this was a deep learning paper. And then within 2 or 3 years, ImageNet was pretty much solved. Now there are programs that can recognize all the categories in ImageNet as well as humans. Okay? So you don't need to know this stuff, but this is how numbers looked. Let's see how the results looked. So here are, this was, if you got it correct in the first 5, if your first 5 guesses were, you got it correct in your first 5 guesses, then it counted as a correct answer. Okay, so this is some of the difficult images, and you can see on the bottom row there are some cases where it wasn't it wasn't as successful, but that had to do maybe with what actually the humans decided that this image was about. So this was called a cherry, but in fact the network said that it was a Dalmatian, which is not a really bad answer. Anyway, so this is a parenthesis. This is just to give you context of how these algorithms work. Okay? So in 2015, when we started doing this work, we thought, okay, well, we will create something similar. We will train deep learning networks on these digital histopathology images. But there was one, let's say very little problem, but in fact it was a very large problem, and how large you will see in 1 minute. Okay? So, out of, we started with brain cancer biopsies. So, if the doctor is afraid that you have brain cancer, they go with the needle and they take a tiny little sample. Okay? That tiny little sample becomes a gigapixel image. Okay? Now, the images of ImageNet are 350 by 500 pixels. That is an ImageNet image. Can you see how it compares with the slide? All the deep learning networks can work with images of this size. Okay? This is what the current technology, what drives face recognition on your phone, what drives a lot of the computer vision applications that you see everywhere around you used. So this technology could not deal with this size of the images. If you tried to scale up and create a network that would take an image like this, probably you couldn't even build a computer that could handle it, that could do all would have enough memory and to store all the intermediate stages, not to mention do the computation. So it definitely could not fit into a GPU. So now I will tell you how we addressed these problems, and I will give you some examples. So these are all images of biopsies. Okay. They are not very interesting maybe to look unless you are a pathologist. My other doctor friends tell me that even in when they're training, they're kind of like weird medical students go to study pathology. But and then they spend 4 years of their lives looking at images like this and learning to read. Okay? So these are very, very highly trained people. Okay, please don't tell my collaborators I said that they're weird. But because they're a little weird, they're really good at working with computer science. Okay? So this is what the images look like, and this is what we can analyze. So, starting with this image, we can ask questions about what is the cancer type. Okay? What is the grade of the cancer? For those of you that have had family members who have had to deal with cancer, you hear about stage 1, stage 2, stage 3, stage 4. No matter what the cancer is, when you hear stage 4, you know it's going to be fatal. Okay? So at this point, for most people, when they hear it's stage 4, basically it's a death sentence. So this is life and death stuff. Getting it right, diagnosing it right, is really important. Predicting what are the chances that the patient will survive. Okay, this is another question that we can ask. Okay, why is this important? Based on these chances, doctors decide on different treatments. Okay, so we decide this on all of the biopsies. Then we can go and look at one particular sample of the biopsy, a little patch. Okay? So I'm going to take a little, as I've been speaking, I will be calling this either a window or a patch. Okay? I go taking a little window, and now I can ask what particular classes of structures of tissue are here. Now things are getting technical. Even after working on this for 4 years, I still can't really tell what a pseudopalisade is. But or if there are tumor infiltrating lymphocytes, this is a very important question because right now the most important new development of cancer is what's called targeted immunotherapies. Okay, so you make the patient's immune system fight the cancer. But what carries the immune system response is the lymphocytes. So seeing whether the lymphocytes are going into the tumor is a very important characterization to know what's happening. Okay? And then you can go even further in and you can look at individual cells. Okay? So we're talking something about going from the original to maybe 5x to 5 times multiplication to 20 to 40 times multiplication. Now you can see individual cells. Now you can see the cancer cells versus the healthy cells. You can see the nuclei. You can count them. You can find their shape, if they're normal, if they're abnormal. Okay? The pathologists don't care as much about this. The researchers care more about it. Okay? So trying to figure them out and segment them and find the boundaries. Okay? So this is another question that you might want to ask. Okay? So you can say, are the cells dividing? Okay? That's called mitosis. Cancer cells divide really fast. So you want to see if this is happening. What is their shape? This has different meaning and so on. Okay? So that gives us a number of different tasks that we might want to solve. And of course, we're not the only ones. This is some of the early work that happened before deep learning came in. And there is now a lot of work that's out there. This is actually a pretty exciting area of research for any of you who are looking for interesting topics that you might want to study later in the future. That's kind of an interesting area of research. Okay. So I guess I'm explaining things a little bit too much, so I won't have too much time to talk about everything in a lot of detail, but I will at least try to give you an idea. So some of the first work that we did was this idea of figuring out the cancer subtype for the brain. Okay? I've already talked about this. Well, so as I've said, you can't put the whole image onto the CNN. So what do? Well, you go and chop it up, not physically, okay, but digitally. You chop it up into little windows, and you try to make a decision about whether there is what kind of cancer it is and so on. There is a complication. Imagine that I'm looking at this room and I say, well, this room has high school students, okay? And I start looking at each of your faces. Well, some of your faces are clearly high school students, but the others are not, okay? So, since I cannot look at all of the windows, there are too many, and I have to pick and choose. What if how do I make the decision? Some of them tell me, well, it's a high school student. Some of them tell me it's a professor. Some of them tell me it's a university student. How do I decide what it is in the end? Right? I need to give one label. And what I know for truth is what the pathologist said. And the pathologist didn't go and tell me what each of these windows are. The pathologist looked at it for 10 minutes and said, grade 4 tumor. Right? So that's all I know. So how do I train my algorithm to predict what the pathologist said with all this conflicting information that I get from all of the windows? Okay, so what you do is you try to find, when you're doing this training, when you're adapting your neural network, you're trying to find what are the patches that are actually going to help your network guess correctly, discriminate correctly. Okay? And you train only on those. Okay? So I will pick this guy who looks like a college student, this guy who doesn't look like a college student, and I'll say, well, a high school student, and I will say, okay, train on these two guys until you learn to predict high school students. Okay? So at the same time, once you have done that, you've trained the CNN once, you can now go and try to look at the rest of the image to find more discriminative patches, and you keep refining. It's a back and forth process. Okay? So you do the training patches, you train the CNN, you identify the discriminative patches, and you keep doing this until you get a probability map, which we will do some smoothing on just to help us with this, to figure out, that tells us how likely each patch is to predict the correct answer. Okay? And then, based on the probability, you will just decide, well, anybody who has probability above 60%, 70%, you decide, I will call them a discriminative patch, and I will keep them. And then I will do the training patches again. This is actually a well-known machine learning problem, which is called expectation maximization. It would probably take me the rest of the lecture to explain it here, but it's a pretty neat idea. It is a mathematical formulation of the chicken and egg problem. Okay? So, you guys know this? Is the Koreans have chicken and egg, or is it something different?
It's the same. We call it
chicken and egg. Okay. So this maps exactly to this very well-known problem in machine learning. And then, so we go from patches, and then we count the patches, and we create histograms. Okay? So we create these histograms of the patch level classes, and then based on this histogram, based on the distribution of the high school students, the college students, and the faculty, we are going to decide whether this is a fun group of people or a boring group of people, right? So this is how we're going to do it. There's lots of different ways to do it. There are standard deep learning ways, voting or max pooling, and so on. We chose, again, something that's called logistic regression, which in the end will decide for us how to call these histograms, these distributions, and give us the concept. Okay? Of course, we tested this and we compared this with other methods that were not using deep learning. Guess what? We did better. Otherwise, I wouldn't be telling you about this. What is more interesting, though, is that in a similar study, not with the same images, okay, but in a similar study for this cancer, where they asked 4 pathologists to read the slides, and then they measured whether the pathologists agreed. Okay, that highly trained doctors, they had approximately the same level of agreement. Okay, so what does that mean? That the labels that we were given by the doctors were only about 80% sure that this is what it is, because they cannot agree within themselves. Okay? So, we tried this on a different problem just for the fun of it, and we actually did a good job in figuring out which of these different images of train track needs regret. Okay? So, okay, we can classify cancer types, but how can we help search for the cure, right? That's what's even more important. Well, for that, you want to actually be able to identify structures inside the biopsy. Okay? So so that researchers can see what's happening. It's not enough to just call it something. You need to be able to describe what's happening inside so that researchers can actually look at the different types of cancers, look at how the different structures inside the cancer affect the response to treatment and so on. Okay? So this is what I was talking about in patch classification. And this is where we are. And so what we want to find is these creatures. You can see them with the red arrow. They are called tumor-infiltrating lymphocytes. Okay? As I've said, this is really important. We want these guys. We want to give immunotherapy drugs, and these will be the guys that will tell us whether these are going to work. So we wanted to classify patches to see if they're still positive, and we just said, well, if there's more than 2 lymphocytes in there, then it's positive. If it's 0 or 1, then it doesn't have the lymphocyte. Okay, so straightforward approach would be to run a CNN and then decide whether it's a lymphocyte, and that would be the same as what's called a bag of nuclei representation. That means I throw everything in a bag, and I don't know how they are related. I just learn to recognize if the stuff I'm looking for is in the bag or not. Okay? So I'll know that there's 14 rounded small things, 2 elongated things, 1 irregular. Okay? The problem is that we don't have training data. Okay? In order to do this, a pathologist would need to sit there on the computer with a mouse and circle these lymphocytes for thousands of patches. Okay? In the United States, these doctors are paid about $200 an hour. Okay? Plus, it's really boring work. So you get them to do it, but they will do 1,000, 1,500. That's it. We need much more. So we instead tried to do what is called unsupervised learning method. Now, what's unsupervised learning? It says that you don't tell the computer what is the correct answer. Okay? You just give data to the computer, and you hope that the computer will actually figure out how to describe things so that the good stuff goes on one side, the bad stuff goes on the other, or the reds go on one side, the blues go on the other. Okay? So this is a very kind of wavy way to think about it. What we wanted to do is to, without providing much supervision, we want to be able to give an input patch like this, reconstruct a patch like this, where we lose all the irrelevant information, and we're left only with these dark things, which is what we want to actually study and count. Okay? There is a technique in deep learning that is called autoencoders. It does exactly this. It takes in images and learns how to reconstruct them. Okay? So imagine a network that starts analyzing the image, learns features, breaks the image down to these features, then learns how to combine them, ends up with some code for the image, and then this code goes into another network that recreates the image. Okay? So you break it down to some series of symbols, and then you learn how from these symbols to reconstruct it. Okay? All of you at some point when you were kids, you took apart your favorite toy and tried to put it back together. Okay? And maybe the first time that you did it, you weren't that successful, and you had a bunch of pieces that were left over, and the toy wasn't working because you couldn't put back the wheels on, or I don't know what. But then took it apart again, and you tried a little bit better, and you put it back, and now you had fewer pieces. So you thought, okay, I'm doing better, and you tried again, and fewer pieces, and I'm doing better. It is a very similar process what you will do to see if you are encoding and decoding the image. Okay, you're comparing the final image with the original image, and you try to make them as equal as possible. There's nobody that's telling you this is this, this is that. You're just saying the output of my network needs to be the same as my input. The difference needs to be as little as possible. Okay? This is a general technique. It's called autoencoders. And in this particular case, we wanted sparse autoencoders, which means autoencoders that have only worked on a few parts of the image and didn't care to reconstruct the rest. Okay? This was important. Why? Because these are really large images. If I'm going to try and learn a representation for everything, okay, then it will require a lot of data, a lot of computing power, and that would be a problem. So instead, say that I'm looking at this room, and all I care about is the water bottles. Okay? Sorry guys, I don't care about your faces. I care about the water bottles. So I train my autoencoder to reconstruct the water bottles really well, and the rest, no, I don't care about questions. I don't care about the error. I don't care about anything. So this is how it would look when we don't do a very good job at reconstructing, and but we're on a budget, so we do really large windows, and then this is how it would look when we do high resolution. Okay, and this was some work that we got recently published, where we had this sparse autoencoder that we were able to train and do this work, and that was we were able, with a lot of more layers, to characterize the tumor infiltrating lymphocytes. Okay, and again we did quite well, and this allowed us to do very large scale field map prediction. So we were able to run it on a lot of slides. Why? Because this was we're able to do it fast enough that we were able to run it onto a lot of slides, and we produced maps of where there is high concentration of TIL maps, of TILs for 5,000 slides in 13 different cancer types. That's humans could not have done this. Okay, and then biostatisticians, these are the people who analyze this kind of medical data, were able to actually show that the kind of the organization of the tumor infiltrating lymphocytes actually predicts survival. Okay, so this is actual real science result that came out last year, and it's kind of important. So I think I will skip this one because I have 50 minutes, right?
Take your time. If you want it longer, you can go ahead.
I don't want to bore everybody, so I do want to talk about the penguins a little bit, but okay, let me just fix this. Okay. So what if we can actually count nuclei, classify nuclei? Okay, we can now talk about segmentation and so on. Okay, so again, there's millions and billions of nuclei in those images. And they're real different. They look different for each different tissue. They are different, and you need pathologists to annotate them. I'm sorry, guys, I can't I can't pay you, what's a good price for to pay a high school student here in Korea? For a high school student to do a job for you.
Okay.
$50 an hour? $7, but okay. So I can't give you guys what would that be? 7,000 won an hour?
It was actually 8,000 something.
8,000. I can't give you 8,000 won an hour to sit there and draw circles. Why? Because you don't know. First, I would need to train you, and I would train you to just do only one time, because you don't know all the others, right? And so on. So you need pathologists, and they are expensive, right? So what we tried to do was we tried to actually use very little pathologist data, very little human annotation. You can't do it without any, but we try to use as little as possible to learn how to segment this nuclei. Okay? So, and there is another very popular technique in deep learning, which says that if you don't have enough data, like real data, you synthesize them. Okay? You use computers to generate, which works really well, say, if you want to learn a computer to learn how to drive a car, right? Because you learn it on, you create data on the computer on a driving simulation game, okay? And you run it for many, many, many hours, and you have lots of images, and then you can use those images, right? But there's no computer that knows how to synthesize biopsies. Okay, so we were the first to attempt this. Okay, and this is a technique that's called data augmentation. Okay, very important technique in deep learning, in being able to solve problems. So, as I've said, it takes hundreds of hours at $200 an hour to label just a few types. And the moment you train a model on just a few types and you try it of cancer and you try a different type of cancer, it won't work. Okay? So we try to synthesize both the texture and the ground truth structure in an unsupervised manner and have a model that can generalize on every tissue. Okay? There is a famous class of algorithm called the generative adversarial networks. Okay? So this is a game. Now you don't have one neural network, you have two. One network generates images. Okay? Lots of images. The other network is one of those recognition networks. It's a discriminative. You give it some real images, and then you give it the fake images, and it tries to say, is this real or fake? Okay? Imagine that you are trying to become a good, not that I suggest that as a career path, stick to this, but imagine you are trying to become a very good art forger, okay? And you are trying to imitate the style of a particular painter, okay? Can I have a painter? Let's say Van Gogh, okay? So imagine you are trying to imitate the style of Van Gogh. You paint something, okay? You take it to your mom. Your mom maybe will say, oh wow, is this by Van Gogh? Okay, you give her a photo, not the real painting. She wouldn't do it at her painting, but by Van Gogh. Okay, so then you're like, okay, I'm good. Okay, so now you're getting a little bit bolder, and you take it to your art teacher, and your art teacher will say, who are you trying to fool? Okay, so you keep trying, you're becoming better. Okay, and at some point you become so good that you will fool your art teacher. Okay? And then you will go and try an antique collector or a painting collector, right? A painting expert. And now you try to fool that person. But what happens is that as you're giving better and better examples, okay, the expert becomes better also in learning the fake versus the real. Why? Because as the fake gets closer to the real, the distinction becomes much finer. So you learn much more fine distinctions. So this is an adversarial game. Okay, both are trying to win. At some point, there is some kind of a balance. Okay, the generator can't really get much better. The discriminator can't get much better than you saw. So this is the standard technique. It's called a GAN. Again, very, very popular. So this is what we try to do. You start from any input. You input a number of real patches, and now you start generating synthetic patches. Okay? And this is our generator and discriminator. The generator generates these patches. The discriminator compares them to the real patches and tries to become better at recognizing them. Okay? And they play this game that we are doing. We need two kinds of examples. We need real patches and we need masks. Masks are the boundaries. What tells you this is cell nucleus, this is background. Okay? And what we do is that we start with a possible mask, and then we synthesize a patch out of this mask using the GAN, and we weigh this resulting patch accordingly. Okay, so I'm going to skip the details. It's a rather complicated system. It would take me about 10 minutes to explain, so I'm skipping this. But what I am going to say is that this wasn't working too well for us. And what we figured out was that because there is so many different things that you can generate, some of them were pretty bad. So, and then we were training classifiers. We were feeding all this data to something that was trying to do the segmentation. So what we learned to do is we actually learned to estimate for each patch that we generated how good it is. Okay? And the good ones, the ones that really look more like real patches, we just gave them higher importance. Everybody got a little bit, but we gave these guys higher importance. So this was the big thing. I'm skipping this. This is how good our results were. Okay, so you can see how we were able to generate all these different patches. Interestingly enough, the pathologists that we work with, the night before the submission, the PhD student Blackout sent him a bunch of those patches, and he asked him to guess if they're real or fake. We got him 50% of the time. Okay? In all fairness, if he had to do it a second time, he would get them. He would know what it is that the fake ones look like, and he would get it right. Okay, because he would train himself, just like this discriminator. And the truth is again that pathologists, they don't look at patches like this. They come from a bigger image where they have a context, and that helps them explain things, right? So, but anyway. We were also able to do this with numbers and so on. And again, I did really well with a little bit of real data and a lot of synthetic data that seemed to work the best. So label super resolution. I promise this is the last thing I will talk about because we'll have to time this for another topic. So here's another thing. This works for satellite images also, so at least I think we've talked about satellite images. In case you guys didn't understand this from this talk, these images are huge and it's really impossible to annotate. Okay? Which means that it's impossible to get ground truth labels for every pixel at high resolution. But it's much easier to annotate at low resolution. Okay? So how can I annotate in low resolution and use that to help me to guess what things are in high resolution? Okay? So what happens is we can assume that the low resolution labels are correlated with the high resolution labels. Okay? Which means that if I see a low resolution label, imagine on a satellite image, if the low resolution label shows something that looks like a city, then if I go to high resolution, I expect to see a lot of cars. If it shows something that looks like a forest, I expect to see a lot of trees, right? So there is some correlation. There is some connection. So you could say, well, again, talking about the stills, if I have a low resolution label that says that the probability of having tills in this patch is 80%, then I can force my algorithm to expect about 10 to 20% of the pixels to be lymphocyte pixels. And we can do this using KL divergence, standard metric. It tells you here is one probability distribution, here is another probability distribution. How far is it? Okay? We use KL divergence. So I'm skipping this, but let me show you, apart from tumors, how you can do, for example, here is a satellite image where the labels that we had are the ones over here, where they might be telling you it is impervious surface. That means it's something that is covered. It can be covered by road or by house and so on, or it is grassland, or it is water, right? And then you can try to identify houses and trees and roads. Okay? So this is how well it will do by doing this. All you need is to do the correlation somewhere. For a small part of your data, you need high resolution annotation and low resolution annotation. You learn the relationship and then you apply it elsewhere. Okay? Let's skip all the details. Here's again some of our images. Future work in all of this, we still want to reduce the annotation burden. This is the big thing that we're trying to do. So some things that we can try. There's these techniques that are called few-shot learning methods. What you would like to do is that there is hundreds of years, tens of years, about 100 years worth of pathology reports. The last 30 years of pathology reports are digital form, right? Since the 90s, the doctors have been typing this stuff up. Can I take what the doctor wrote and imagine what the image looked like? Okay? The doctor writes all the descriptions. They write about a page for each case that they deal with. Out of this page, can I generate more training data? Okay? There is lots of high performance computing questions here so that we can run it on very large database. There is questions about how experts can make decisions on this. Where it is that experts look. Why do experts make different decisions, different diagnoses on the same image? So studying their behavior. And in the case of satellite images, how do we do temporal modeling for the change of distributions? I'll stop here and I'll take some questions.
So, do you have any question?
Thanks for your talk. In your tumor classification work, do you take your patches to be overlapping like the general CNN patches or discrete non-overlapping ones?
Well, we have tried everything, but we don't necessarily take all of the patches.
Okay.
Because there's just too many.
Right. Then how many of those patches are positively labeled?
In the first one.
Yes.
None.
None.
This is the whole problem. All we know is that some pathologist looked at it,
right,
and gave it a label.
Yes.
And then we will count the patches, and we will see what are the patches that actually make the algorithm change its prediction.
Okay.
These are the important patches. We will keep only those patches.
Right.
And we will, out of the histogram of those patches, we will make a decision. Okay? Any more questions?
So what are the size of the patches? Like?
They were 100 by 100 pixels.
Okay. So and do you use like ResNet or that kind of models to?
We had back in 2016 we used I don't think we used ResNet.
Okay.
I'll have to look back at the paper to remember, but it was a simpler architecture.
Okay. Professor, I just wanted to ask, what sort of accuracy was it? Were you using precision in the papers?
This was we were looking at the percentage of getting it correct. Okay. So how many of those that were category 4 we guessed as category 4.
Any more? So let me give you another question. So you said the context-aware analysis. Right?
Yeah.
The doctor's not looking at just the picture, but they're zooming through and make a decision in time series.
Yes.
Or zooming through. How do you guys approach to solve that?
Well, we have some ideas. They include attention. Okay. We're trying to get money for those.
Okay.
So I'll talk to you afterwards.
All right. Thanks very much. We'll take a 10-minute break and.